Thanks, everybody. All right, so our next speaker, Terry Combs, is going to be joining us to speak about sustainable beekeeping through selective stock improvement utilizing natural selection. I have become a big fan of Terry, and I like to speak with him as often as I possibly can to learn from his experience. He's got a lot of good things to say, which is part of the reason why I've invited him to speak here tonight. And I feel like what he has to offer is going to be worth the wait. Terry Combs is a 100% chemical-free beekeeper when it comes to Varroa control in his apiaries. He has been keeping bees for 56 years. He has the distinguished honor of holding a Boy Scout beekeeping badge as a youth, which entails many rigorous requirements prior to even receiving bees to keep. And though the badge is no longer part of the BSA program, Terry continues his desire to this day regardless. Prior to the 1996-97 season, Terry was running the chemical treadmill. Starting with 83% loss in the spring of 1997, Terry managed through the process to achieve 0% loss in the spring of 2018. Achieving an average of 3% colony loss during this time, his experience has proven effective in developing a practical, hands-on approach to beekeeping in the treatment-free apiary. Terry teaches 4 beekeeping classes on different subjects, 2 insect classes, 1 for adults and 1 for younger people at a local college. He frequently gives presentations for youth and the public on bees, beekeeping, spiders, and insects. And in 2020, he furthered his beekeeping education by becoming a University of Montana master beekeeper. Terry lives in Keysport, Illinois with his wife, Debbie, 2 cats, a tank full of guppies, and about 3 dozen beehives. With that said, I am going to ask you to unmute Terry. Let me know when you're on. We got audio.
Can you hear me?
I can hear you.
Hello, James.
Hi, good to hear from you finally. It's been a long day. I can't wait to hear from you, but at the same time, I'm ready for a nap.
Yeah, I know the feeling.
Yeah, I am going to have you start your video. If we have any issues with your buffering and whatnot, we'll go ahead and discontinue the video so the audio means it stays intact.
Okay, are you running it on your end or am I?
Yep, I'm gonna run the slides for you so that way you can go through your presentation and I'll be at your beck and call here on the slide change.
Okay. And you'll be able, James, to move the frames as I need it.
Yep, that's right. Yep.
Yeah, I will prompt you.
Okay, great. All right, give me one moment here. Zoom is not cooperating. Just give me one moment.
I'm a patient man. I'll put it this way, James, I'm retired. I got all the time in the world.
All right, sounds good then. Just as long as you're not charging me by the hour.
No.
Okay, I'm going to stop sharing the screen here for a moment and then re-share it because it is, again, not cooperating.
Okay.
There. All
hey,
right.
There we go. That looks familiar.
Yes, this is your presentation. Terry, you may feel free to start when you're ready.
Okay, well, I'd like to welcome everybody and thank you especially for going through this whole day and staying here to finally listen to me. I do recommend that maybe you have a piece of paper and a writing implement with you, maybe jot a few notes down. Like James said, I've been keeping bees now for 56 years. Back in 2020, I enrolled in the Master Beekeeper program at the University of Montana. Beekeeping is a lifelong educational experience. I've decided it never stops. I'll be quite honest, the Master Beekeeping course, I really didn't learn a whole lot that I didn't already know after 50 some years, but I did learn more. For one thing, they delved into anatomy, internal anatomy and external physiology, which I was lacking on. I picked up quite a bit of knowledge there. The most interesting thing that I learned during that course has to do with this slide here, bee flight. We had to review a paper about how bees navigate, and it was mind-boggling to me. It's kind of like they run a video in both sides of their brain with each eye, memorize that and kind of rerun it on the way home. I don't even begin to say that I fathom everything that was in that paper, especially when you consider they figure in the angle of the sun and everything like that. But as Mr. Spock once said, it was absolutely fascinating to delve into their ability to go out there, find something, and get back home. I value education very much in beekeeping. Unfortunately, a lot of what I see that is passed off as education nowadays falls far short of the target. I most value the education I got when I started beekeeping through the Boy Scouts, because I got connected with the ultimate mentor. And one thing about Edgar, the guy that mentored me, he wasn't going to let me off of any of the requirements. He produced basswood section comb honey, and I had to learn the whole process, which I kind of, at the time in my youth, felt was sort of boring and a lot of work, but I appreciate that he kept my nose to the grindstone. Let me start off here by saying you're going to hear some things that you may not have heard before, some new ideas, different viewpoint, but let me make a case for different viewpoints. Controversial viewpoints are good for any field. It spurs inquiry on all sides of a question, as differing and opposing viewpoints will argue to counter arguments and evidence from the other side. This generates new arguments and evidence and bolsters cases and viewpoints. And this goes on. And it's especially valuable in the field of research. And one thing about researchers and scientists, they can have these arguments without bashing each other. One other thing I'd like to define here, since I've seen it in a lot of these presentations today, it's the first word in mind, sustainable. This comes straight out of a biology text from the college where I teach my classes. Sustainability requires that we respect the natural world and the natural processes that sustain life, not just our lives, but all other lives. And it requires that we reduce our inclination and orientation toward short-term personal gain. We don't need any more doom and gloom about the problems that we're having with bees. What we need to do is aggressively pursue more knowledge and be willing to work towards a long-term sustainability for all. That's my definition of sustainability. Next slide, please. Are you there, James? Okay. We humans have set out to try and control a part of nature, and boy, we're finding that there are problems with that. And the reason we're having problems is that bees are a part of the natural world. They are not pets. Any of you that have followed anything I've ever written in the bee journals, I've had quite a discussion with Jerry Hayes, who wants to say bees are like pets. Nothing could be further from the truth in my mind. As Rachel Carlson said here, nature does not exist for our convenience. We are a part of it. It is a part of us. We try and control it, but oftentimes we find that our methodology does not work. Natural selection has its own methodology to solving problems. Bee breeding has, up to this point, pretty much been in the hands of commercial queen producers, researchers, and scientists. Some of these people are not even beekeepers. And what they've selected our bees for and bred them for, although I use the word bred lightly, they've selected for gentleness, low to no swarming, honey production, no propolis, disease and pest control has been chemical. And this goes back to what I said on the last slide. It's part of human nature. When we have a problem, we want some solution that will solve that problem as quick as possible. And so when Varroa came into the US here, we all, and I did it too, we all jumped on the chemical treatments that were offered because it looked like a quick and easy fix. Instead, it's been kind of a horror from where I sit. These bees are selected for our human benefit. Let's just be quite honest about it. That's what we're doing. Natural selection in the bees, though, left to their own devices, don't select for the same things that we do. They select for what benefits them, and they've been doing it now for some 35 to 65 million years, depending on who you want to ask, as to when they diverted away from their solitary Hymenoptera ancestors. In starting a program like I'm going to lay out here, one thing to remember that natural selection is on your side if nobody else is. Natural selection is trying to solve the bees' problems, and it's a powerful ally, and it needs consideration. We've kicked the can 30 years down the road with the chemicals and everything else, and all I have to do is look at the yearly bee losses every year, and I can tell you the chemicals aren't making anything better. These laboratory bred queens with their special jacked up traits, they're not making anything better. Nothing's making it any better. We keep having worse and worse losses. It's time to stop and reconsider this and start working, like several of the other speakers have said, start working with your bees and what they're trying to accomplish. There are many what I call aberrations in beekeeping that disturb and conflict with and undo natural selection's progression and benefits. It's time we start taking advantage of those and stop interfering with the things that natural selection is wanting to accomplish. I hope I live long enough to see the day, because even our researchers have said this. Eventually, there's going to come this population-wide resistance with bees if we wait long enough. Unfortunately, a lot of what we're doing is kind of delaying that process. I do hope that I live long enough, though, to see that day come when naturally on their own, bees pretty much everywhere have developed resistance to Varroa. One of the reasons I hope I'm around is because I can't wait to see all these researchers and bee experts say, well, see, we finally got here. It was a struggle, but we finally got here and we solved this problem. Because I'm going to laugh at them and tell them, you didn't solve a thing. The bees solved this problem. You only stood in their way. There are a rainbow of opinions and thoughts and beliefs on how to proceed with this problem. Like I say, I started out with the chemicals and I didn't like what I saw. I was getting nowhere. It was a merry-go-round of colonies die after a couple, 3 years, replace them with new colonies, and the same old thing. And the winter of '96, I had 100% lost everything, and decided that spring when I ordered a couple packages. I'm going to try something different and see if maybe putting the control into the hands of the bees might get me somewhere. So next slide, James. Let's first take a look at what's going on currently here. And I'll call your attention to the top right hand of the slide. These two papers that I've referenced here. Natural Varroa Mite Site Surviving Apis mellifera Honeybee Populations by Barbara Locke. Anybody that does not think that resistance in bees and bees that can survive on their own without treatments is an impossibility needs to read this. Because not only Barbara, but Tom Seeley, LeConte, researchers all over this planet are seeing not only colonies out in the wild that are surviving without treatments to Varroa and surviving with Varroa, but managed colonies. A good example is Tunisian beekeepers. They were too poor, kind of like previous speakers. Jamaican beekeepers, they couldn't afford all this mite chemical treatment. So they just plodded on and their bees developed resistance on their own. Now, once you get people to understand that this is possible, knowing beekeepers like I do, they're always going to find something to complain about. And one of the things they complain about is, well, your bees don't make honey like my bees did. They've always got some excuse why it doesn't work for them. Well, the next paper down below is another recent paper that shows that local honeybees that are adapted to an area can have lower pathogen loads and higher productivity compared to the non-local bees that you bring in packages, the queens you ordered. Allowing yourself to create a stock, a flock, a herd, whatever you want to call it, of your own bees and letting them adapt to an area will bring about beneficial changes. It can be done. Now, one of the problems in the face of the fact that we've got proof that bees are surviving Varroa without chemicals and doing quite well are the mixed messages you get in the beekeeping world. So let's move down to what Jerry Hayes said back in the June issue of Bee Culture. Now, this was in answer to a reader's question about the bees in her area that other beekeepers bring in affecting hers. Well, to be quite honest, he never answered a question, but he ended up his talk with this quote I've got here. We in the US have such high losses every year because most hobby beekeepers don't know how to get past buying packages and they blame somebody or something else for their losses. Okay, well, there's some truth in that, Jerry, but you'd go back 6 pages earlier in that magazine and what do you see? You see Jerry walking away with his spring package. That's a mixed message. It's just like I go to beekeeper meetings and I've heard this a gazillion times. Ask 10 beekeepers the same question and you'll all get 20 answers. And they sit around and joke about that. And every new beekeeper that I've ever asked about that statement tells me that that does nothing to help them. There are too many mixed messages. In fact, there's outright denial down there. I got an email from a guy named Ben a few weeks ago. He'd seen a previous Zoom conference that I was on and decided to explore the route of trying to breed bees and get them away from chemicals. And he went to his local association and was laughed and basically almost thrown out of there, saying it can't happen. It takes thousands of years. Folks, let me tell you, if you ever walk into a bee association and they tell you that, I'd turn around and walk out. You're not going to get anywhere with closed minds. We need people to open their minds, and most of all, we need people to try this. This has been talked about far too long. It's time for some action. I'd also like to say, when I captured this picture, and I'm not here to bash Jerry or Jim too or anybody. I'm just showing you the mixed messages that are being sent out there. In fact, I got permission from Jerry to use his quote and his picture. He said, go ahead if it helps. Well, it does help illustrate the fact that you're sending mixed messages out there. Jim, too, the guy in the upper photograph there holding a camera, I think, in one of the recent issues of bee culture, he noted that he's getting older like I am, and he's going to smaller hives. Good idea. But then he said, I don't know why I need to worry about getting the best queens anymore. I think mediocre queens ought to be just fine. Well, if you're setting out to breed something, folks, any breeder, whether plant or animal, will tell you that you don't deal with mediocre specimens. Like I say, some of the messages out there to me are just nonsensical. There was an American Bee Journal article recently from someone here in my state who I know, and he's a commercial beekeeper, said that it's impossible for bees here in the US to do any adaptation or evolution. Nonsense, folks. Every form of life on this planet is subject to adaptation and evolution. We can maintain locally productive resistant bees. I've been doing it now for 26 years. No chemicals, no replacement queens. I can take the queens that I have in my colonies right now, and through the records I keep, trace a straight line back to 1997 when I ordered those 2 packages. Like I said earlier, the losses that we keep having every year and the fact that they're not going down, they're actually going up, tells me that chemicals, laboratory created queens, they're not solving anything, folks. You need to look elsewhere. Next slide, please. Let's not forget either what we human beings are capable of. We put a man on the moon. We're capable of all sorts of things if we really want to do it. And I think that's part of the problem here too. People still are stuck in that I want something I can pour out of a bottle, or I want a queen that I can buy and stick in my hive. It's going to solve all my problems. And you're not going to find it. However, my copy of The Hive and the Honeybee, 78 copy, has a quote, or I have a quote here from Mr. Kale and Mr. Rothenbuehler about this situation. And what they said was, we can have greatly improved bees if we want them sufficiently. Well, I've heard a couple of the speakers today say the same thing I'm thinking. Everybody, if you ask them, yeah, would like to get away from treating their bees and have bees that are successful and improved and get away from some of these problems. So I'd say it's not a lack of desire on people's parts. Maybe they don't desire it sufficiently though. But I think a lot of the problem is what they go on to say, which is it's a matter of devoting time, intellect, material resources, the necessary research and development. In other words, there's some work you got to do, folks. I'm not going to give you an easy solution to this. There is none. Part of the problem is our own efforts. They've stood in the way of bringing about the natural equilibrium that usually comes between a host and its parasite, and will someday with bees. So you may be asking, can we really do something like this, aid bees in developing resistance and better health? Yes, we can. And one other thing, a lot of people think this is some new idea, resistant bees. No, that message has been lost in the past. Next frame, please. A little bit of background here. This quote comes from a paper that Oscar Park read to the Association of American Entomologists meeting in St. Louis back in 1935. He was talking about resistance to American foul breed. And what prompted his study in this paper was the fact that beekeepers back in the 20s and 30s, some of them were saying, hey, I've got bees that apparently resist this disease. They've had it, they've recovered, they're doing fine. Back then, it was thought that having American foul brood was the kiss of death. I think in some ways that attitude still pertains to today. So these Park, Paddock, and Frank Pellet, 3 researchers, there's a 4th guy in there. I think he was with the USDA, which got involved in helping propagate the resistant bees. But they set out researching this, and they found that not only does American foul brood resistance exist in bees, it's also inheritable. And that's why Park went on to say that if resistant bees exist, we cannot begin too soon to propagate them. If they don't, the sooner we find it out, the better. This argument has gone on long enough. It's time to settle it. So back in the 30s, this was talked about. And in fact, in the 20s, Oscar Park released a paper about breeding for European foul brood resistance bees. So this has been talked about in the past, folks. This is nothing new. There's a lot of lessons from the past in beekeeping that we don't seem to have learned a lot from. To quote Merlin in the movie Excalibur, it's the doom of men that they forget. The Africanized honeybee should have taught us a good lesson about moving bees around on the face of the planet. The evolution of subspecies offers a good lesson in what happens when you don't move bees around and they're allowed to settle into an environment. They wind up fitting into it like a hand into a well-fitting glove. These lessons, they seem to come and go, and you know, the hive and the honeybee, or no, not the hive and honeybee, the ABCXYZ of beekeeping listed September 20th, 1934 as a pivotal day in beekeeping. That's when Park and these guys set out to do this research. Well, unfortunately, I don't think it's all that pivotal because it seems to have been ignored and forgotten as we go for a quick fix in all this. James is setting up kind of a competency course for new beekeepers based around the Boy Scout merit badge, and it caused me to pull out my Boy Scout merit badge because some of the requirements that he'd sent me were worded a little different than my merit badge book had. But leafing through that, I happened to notice that even the Boy Scout manual, the one that I have from '57, lists resistant bees as a way to combat diseases and pests. So let's take a look at how we can possibly get there working with our bees and natural selection. Next slide, please. And what I propose that you use is selective stock improvement. Now, the smarter of you out there, you're going to say, oh, whoa, wait a minute, Terry. This has nothing to do with natural selection. You're back to artificial selection, human-based selection. And yes, this involves some human-based selection, but I'm not going to be selecting my bees for necessarily what I think's best. I'm going to be working with them on what they found that's working best. Good example of selective stock improvement is what fish hobbyists have done with the common guppy, Ocelia reticulata. These are wild specimens that you see in front of you here. You'll notice that the female, the top fish in the top picture, is virtually devoid of color. The males are not all that colorful. There's nothing spectacular about them. These are wild specimens. However, by using selective breeding, we humans have created something completely different. This human species modification, as I like to call it, is brought about by selecting individuals with the best examples of the desired traits, behaviors, characteristics as breeding stock for future generations. And this selective stock improvement works very well in the pet industry, this human-based artificial selection. It is, however, removed from natural selection. Artificial selection relies on the keeper, either the beekeeper, the fish keeper, the dog keeper, whatever, to set the course for things. Bees do not. We cannot take bees and put them in a tank, a cage. They are connected to the wild world and always will be. Therefore, we have to recognize and honor the fact that natural selection is directing them. Even if you direct the bees in captivity, you still have a feral population of bees out there that are on their own, truly in the hands of natural selection. The process and methodology of this utilizes observation, testing, evaluation, selection, culling. By culling, I mean just that, culling out what is not getting you where you want to be. And this happens out in the natural world. Unsuccessful genes get thrown in the trash pile. And in order to cull, let me point out that you don't have to necessarily kill the entire colony. A queen change will change the genetics within a colony very easily. And what we want to do with all this selecting, testing, observation, evaluation, culling is effect changes that are designed to improve and enhance and alter behaviors, characteristics, appearance. But what I think we should be doing with selective stock improvement on bees is helping them along with what is working with them. My definition of a beekeeper is a person who is a provider and an assistant. That's what you are. They know what to do. All you've got to do is provide them what they need and assist them in that. And that's what I'm proposing here, is that you use selective stock improvement to assist them in what they're doing by noting what is succeeding and encouraging it. Selective stocking improvement drives the show circuit for fish. It drives the breeding of plants and animals for our purposes. And one thing I'd like to point out about these people is they are dead serious about this. They are dedicated to it. This is not just some flight of fancy for them. They invest time, money, effort, labor into this wholeheartedly. And the fact that there are fish keepers out there that can do what they do, I know there have to be beekeepers out there that can go down this path also. And I'll tell you something, I'm going to be real blunt about it. If I consider the efforts of the people that breed these fish and the efforts of bee breeders, folks, the fish keepers make you look ridiculous. These are serious aquarists, serious fish keepers. And actually, that's what I'm looking for in this. I don't want somebody who's going to just plop a beehive in the backyard, walk away from it and think, well, the natural selection will take care of them. No, that's not what I'm looking for, folks. I'm looking for some dedicated beekeepers, and I know they've got to be out there. Next slide, please. Looking at these fish, you'd hardly even recognize that they came from what was on the previous slides, but this is what these people have brought about. These are, some of them are show specimens. They are show guppies, a product of this selective stock improvement. If you look at them and think back to the previous frame, these fish here are large, robust. They come in a rainbow of colors and patterns, even the females. These breeders have been able to breed some color into. They have unique and spectacular finnage. They are healthier and longer lived than the wild specimens. They have excellent disease and pest resistance within their environment, which are those tanks. They are pets. What would happen if I took these fish out and released them in Trinidad where the puppy occurs naturally? Most of them wouldn't last a day. Any predatory fish seeing these flashy colors and showy fish would gobble them up. Those that manage to survive that are next going to be attacked by diseases and parasites that they've been free from in these fish rooms that these people have. And I know you all realize, like I do, there's a lot of expense in beekeeping. These people too invest a lot in these fish. One thing you don't see with these tanks here, aquarists learned the value a long time ago of the food that they feed their fish. And although many companies try to provide all sorts of artificial food, they found out a long time ago the best foods are the natural live foods that fish eat. So in addition to raising these fish, most of these people also raise other forms of life, microworms, tubifex worms, daphnia, things like that, brine shrimp, to feed these fish and bring them to these healthy conditions. Are you still there, James?
Yep, we had lost your video, so we still have your audio apparently.
Okay. Well, I'm still talking on that screen, so do what you need to do.
Nope, you can continue.
Okay. Like I say, these people are dedicated to the selection process. Some of them are also dedicated to something called species maintenance. And there's a parallel to that in the beekeeping world. European beekeepers have started maintenance programs for some of the endemic bee species over there. And once again, those people are dedicated to it in the fish world. Lots of times, or a lot of these people who are engaged in the maintenance programs are the only source anymore of the fish that they are breeding and raising. They've gone extinct into the wild due to the pet industry pulling them out of the wild and due to the removal of their natural habitats. So we have fish keepers out there who actually keeping fish still in their tanks that out in the wild are now extinct. And these people are all very much dedicated to it. Let me say something, and this kind of ties in with the sustainability thing. The beekeeping world has its Reverend Langstreth, who is widely recognized as the father, the grandfather of beekeeping. The fish world has a similar name, and that's Dr. Herbert R. Axelrod. In his book, Breeding Aquarium Fishes, Volume 1 in 1967, in the introduction, he said this about fish keeping and breeding. He said that the most interesting and rewarding part of fish keeping and breeding, even greater than any financial reward, is the observation of the behaviors, the maintenance of species, the creation of new life, color, patterns, and finnage. That gives far more satisfaction than any money to these people. And I know that for me, 26 years now of no chemicals and a maternal line of bees to where I don't have to get queens anywhere else brings me a lot of satisfaction. And another parallel in the fish world, most of the species that have been bred were first bred by just plain old hobbyists. A lot of them were smart enough to take notes of the whole process when they first bred something, and they would send it in to the trade magazine, Tropical Fish Hobbyists, and have it published as the first person to have bred such a thing. One thing that I would hope that any of you who go down this path I have, you'll find that record keeping is very important in it. If you do this, and let's say you decide to go in business selling your own bees, you're going to have records that now legitimize what you're saying. Because right now in this industry, there are no controls. I could advertise bees and basically say whatever I want about them and produce no proof and get away with it. If you keep records and can show that over time your bees have less and less mite load, that the colonies are surviving and you have less and less losses, and that they're just as productive as any other bees, if you've got this all written down, I encourage you to submit it to the trade magazines in the beekeeping industry. Because one thing that they can't argue with is the facts of what's happened with someone. And this goes hand in hand with this breeding program. Are you still working on the video?
Yeah, let's give it a shot. I'm going to go ahead and ask you to start back up.
Okay.
You should get a prompt.
You got it?
Yep.
Okay. Next slide. Aha, okay. Before you start down this path, I don't recommend it to anybody who's brand new to beekeeping. You're going to need some knowledge and experience. And these are some of the areas that I think you have to have some knowledge and experience in. Ignore the last one for the time being. But bee biology, what I call bee botany, I got that from Dr. Dewey Karen. I think the location, equipment, management are all very important. And then I'm going to introduce you to some record keeping. And it's important if you want surviving, adapted, chemical-free bees that you turn loose of the chemicals at some point. These things here are necessary prerequisites in your education. One thing that I hope everybody gets out there is a good mentor. I exceedingly value the one that I had and the course that brought it about through the Boy Scouts. I'll say this, the research paper I did with 2 other students for the Montana Master Beekeepers program was about beekeeper education. And I found out that a large percentage of people out there that are entering beekeeping have not spent so much as 1 second inside a hive before they get a hive of bees dumped on them. That should never be happening, folks. You should have an in-hive mentor. And let me make a recommendation that anybody starting out with bees, if you go in to get started with it and somebody wants you to buy bees from them and says they'll be your mentor, they better be getting in the hive with you. Otherwise, I'd dump them and go find someone else. This nonsense of sitting around watching videos, listen to 10 different beekeepers say 10 different things and try and learn something. That's not how I learned beekeeping. I learned beekeeping by getting in a hive with my mentor. Your mentor should be your most valuable resource. And as I've said, as far as education goes, this is my 56th year, and I still continue to educate myself in beekeeping. Now, anyone that's out there thinking, well, what you're promoting though, treatment free, that where do we get that education? There's nobody out there teaching that. That's changed. Also at the University of Montana, Professor Debnan, the guy to the right in that picture of 3 up there at the top, now teaches a natural beekeeping course online. So you can find sources of higher education for this now. and I encourage you to take it. I think Scott did a very brave thing and a very wise thing by starting this course. Okay, and as Edward Wilson said down there, he's one of my favorite entomologists. Teach me, I forget. Show me, I remember. Involve me, and I finally understand. That's what your mentorship should be. Next slide, please. Okay, bee biology. What you need to know there is the life cycle of the honeybee and the life cycle of the colony. And you need to know them thoroughly because this is going to be important when you start making observations. You need to know what they're up to and what they're doing. You need to know how beekeepers in your area interact and manage colonies in your particular locale. You need to know what the bees are trying to accomplish, and you need to know about their development, behaviors, and activities. I always recommend that anybody starting in beekeeping get a copy of The Hive and the Honeybee and read Norman Gary's chapter on the activity and behavior of honeybees. It should do away with any notions that your thoughts are always going to be right and you know what's going on more than they do. Don't ever get that idea. Bee botany, I got this from Dr. Dewey Caron. He thinks, and I feel just the same, I'd kind of been doing it without him having put a name to it yet. But the botany part consists of knowing your local flora, the bloom times, how the season progresses and changes, and most importantly, the nectar flows in your area. Especially if you're considering producing honey, you need to know what flows are the ones that you're going to get a honey crop from. So this is one aspect of your learning that you need to tuck under your shirt or tuck under your hat. Next slide, James. Location is very important. This is a picture of my apiary a couple of years ago when I just made nukes. When you consider a location, it needs to have good forage, season long, it should be varied. If you're going to engage in honey production, you need to have at least one major nectar honey flow. Hopefully there are not pollutants and contaminants, or as few as possible. And hopefully there is enough for all. Something that beekeepers oftentimes don't take into consideration is the carrying capacity of an area. You need to figure in your bees, you need beekeepers within a 3 to 5 mile radius, and remember that there are feral colonies out there also. Carrying capacity is very important because if you overpopulate an area with bees, it's to the detriment of all of them because suddenly they're all competing with one another. And that's not going to go well if you've got more bees in an area than you have resources that can be divided amongst them. Water sources need to be also uncontaminated and create no problems. If you live in town, you need to provide a water source so that they don't go to your neighbor's swimming pool. This goes with swarming too. You need to control that, especially in a town, so it does not create any problems. You wonder why some municipalities have beekeeping ordinances. It's because in the past, irresponsible beekeepers let their bees do things that irritated their neighbors. Not everybody's going to have the same attitude about bees that you do. You'll need plant saps and resins from trees and bushes so they can gather propolis. This is one of their defense mechanisms. One of the things that you'll learn in the natural beekeeping course, should you take it, is that propolis envelope is very important to a bee colony. And I've started doing what Scott suggested in the course, which is roughening the interior wood on my boxes so that they can make that envelope. Accessibility, whatever location you choose needs to be accessible to you pretty much all the time. If you need to get out there, you need to be able to get out there. It needs to be permanent. And the reason for the permanency is this. Natural selection does not just operate genetically. It operates within an environment. That is the other thing that is most important in its workings. And what you want to find is a location where you're going to keep them and they can adapt and evolve in that environment. You need protection. You notice, you look at my layout here, you're looking straight ahead is to the north. There's a line of trees and shrubbery to the hive's back. Off to the left there, that's the west. Once again, they have a line of trees and shrubs to their back. I found, for me, that's the most ideal location to have something back there to protect them from the winter winds. I do not and have never wrapped hives in my area. It's not really necessary, but I do appreciate the wind protection that they get from having something behind them in town. I always suggest fencing bees in. You also do this. I've had trouble out in the wild with vandals, so I try and hide them as much as possible. You'll read a lot of beekeeping books that'll tell you that if you live in town to advise your neighbors that you have bees. I don't do that. And if you're a good, responsible beekeeper, you shouldn't have to. A few years ago, the local paper did an article on me. It showed a picture of my backyard here fence where the beehives are. And about a week later, my neighbor on the other side of the fence came over and said, I never knew you had bees on the other side of that fence. That's what I want. Your layout. You notice I space hives as far as I can. I realize, out in nature, they space themselves up to a mile apart or so. I think in Seeley's latest book, it was like 6.10 of a mile. That's not possible for beekeepers, but I do in the apiary try and spread them out as much as possible. I try and make each hive unique, whether it's painting or configuration. I do all I can to reduce robbing and drifting. I also, Langstreth, I think it was, made a note of this, that bees seem to do best when they constantly have some sealed honey in the hive. And I try to always keep stores in there. One thing that I found is that this helps prevent robbing if they've got plenty of stores, no matter what time of the year. Another thing that I've taken to doing, not in this part of the apiary, but the other half that's not in the picture, I've started planting a lot of small dwarf fruit trees and the like, shrubs. This provides some food for them. But if there's ever a swarm that gets out on me, if you look at the picture here, I've had them get up in that oak tree back there, which is, I don't know how many feet off the ground, but it's out of my reach. And I'm too old to go monkeying around up in a tree like that anyhow. I've found that if you put a bunch of smaller trees in your apiary, odds are they will land there and make gathering them up easier. Plus, like I say, those trees do provide a small amount of food. We're ready for the next slide, James. Equipment wise, I use Langstreth entirely. I'm not about, after 56 years of accumulating Langstreth equipment, going to any other kind of hive. My personal opinion is the Langstreth hive is the best one out there. It's the standard for honey production. It's what I use. Am I so narrow-minded, though, that I would say you can't do this in a top bar? No, you can do what I'm proposing probably in a top bar or any other hive. This is just what I use, and I find it most convenient. I keep anywhere from 2 to 4 nukes per hive. Anymore, about 2 is all I need, because I've got some health problems with my age that's made it to where I can't be lifting, like one of the earlier speakers said, 50 to 100-pound boxes of honey and bees. So I'm going to smaller things now. Dr. Dewey Karen helped me with a horizontal Langstroth hive. And I'm still experimenting around with this to find what works best. But going with smaller hives, I usually only need a couple nukes for divides in the spring because I'm not dealing with some colony with 3 deep boxes of brood. Lord knows how many nukes you could split off of something like that, but my bees, usually 2 to 4 nukes per hive, will allow me to do what I'm wanting to do. When it comes to increase, if you're just starting out, I recommend you only double what you have each year until you find to where it becomes more work than what you're really wanting to do. Personal protective equipment. I've been a coal miner for most of my life, and we had a saying down there underground, safety first. And it's ingrained in me, and I agree with it wholeheartedly. And I agree with it in beekeeping too. You should always have a veil on when you enter an apiary and get in a beehive. Gloves, appropriate clothing, and a smoker. I don't care how gentle of bees you've got, and notice the frame here, or the picture here with the gentleman with the hat on. That's my mentor, Edgar. He's one of those lucky people that kind of had a way with bees and would basically get in there almost nude and work with them. But even with him, I saw the day where he dropped a frame onto a hive. It slipped out of his hand, and the chaos that ensued. As one of the earlier speakers pointed out, you're dealing with an insect that has the ability to kill you. Get enough stings, even if you're not allergic to bee venom, you can get some serious medical issues from it. So folks, I encourage you to be safe out there. One other thing, even if you're one of those lucky people that only gets an occasional sting and it doesn't bother you, allergies can arise at any time in life. And I would hate to think that my being stung all my life would lead me to some point where I could no longer keep bees. So I do recommend people try and avoid stings as much as possible. Other equipment that you'll need for this is whatever else you want to do with your bees. If you're into honey production, you're going to need supers, extractors, and all that. That's not the purpose of this discussion, though. Next slide, James. Record keeping. You're going to find that in any breeding program, this is a necessity. And it's why I feel that a lot of commercial and even sideline and small bee breeders are not truly breeders. They're not keeping any records. Some of the claims they make, they have nothing to back it up. And if you go down this route and you do sell bees and you make claims, you're going to have something to back it up. It makes you more legitimate. Reasons for record keeping is you need a plan in beekeeping. Mike Bush earlier today said when you get in a hive, you should have a plan for getting in there. I agree with that wholeheartedly. Open up a beehive just to see if you can find the queen or something is a waste. It disrupts the colony. They've got to put themselves back together, and you've done nothing. There was no reason for it other than you wanted to go see if you could find the queen. You should have a reason, a purpose, and a plan. This record keeping benefits not only the beekeeper, but also the bees, because these records are going to be what you refer back to when it comes time to make decisions on who stays and who goes. The blue thing there, that 3-hold notebook, is my yard book. It's what I take my inspection sheets out in. Now I've got an example of a breeding sheet there from the fish keeping world, and you can notice how simple it is. I published in the Bee Journal a few years ago a 3-part article, and I put in there one of my inspection pages. And I got to thinking about it once I saw it in the journal that it was rather complicated, and you don't really have to get that complicated. So don't let the next slide when we get there scare you. Records are going to help you with this breeding program if you're in business with bees, selling honey, anything. These records will help you there too, come tax time and things like that. If you're going to be in business, I also suggest you get some business knowledge. I see a lot of beekeepers kind of fumbling around with the business aspects of this because they really don't have any experience of running a business. I even had one told me and said, well, because I'd said something about make sure you pay your taxes. Well, you mean I'm supposed to collect taxes for this business? Yes, I'm sorry, folks, but the government thinks that any money you make needs to be reported. But this record book helps you because when you jot down notes and records, for one thing, when you come back to the apiary next time, before you go out there, you can review those and decide what actions need to be taken. Now, as far as to what records you're going to be keeping in here, production records, behavior records, all sorts of stuff. But what I'm wanting you to concentrate on mainly is the behaviors right now. I want to try and keep this simple, where what you're looking for is resistant mechanisms that your bees are using to be successful with Varroa, and you're encouraging those behaviors and trying to do like people have done with the guppy, take it from this level up to a higher level by working with the bees and what they're trying to do. You can record things like honey and the amount of frames or pounds harvested. You can use percentages or actually count things. When it comes to behaviors, a lot of that's going to be observation and opinions. But let me tell you, those show fish that the fish keepers created, they do all that basically by eye. They're not doing any tests. About the only measurement they make, I think there's a specified angle for the caudal fin, the fin at the rear of the fish. There's a specified angle for that. Other than that, it's mainly by eye, folks. And simple observation can yield a whole bunch of information and knowledge. I'll give you a quick example. Young boy that I knew was interested in ants. And in fact, one of the ants that caught his interest was the genus Polyergus, the slave-making Amazon ant. He'd read a few books about how they make new colonies, where the virgin queens went out, mated, and then found a colony of the ants that they parasitized and crawled in there and killed the queen, took over the brood, and eventually came to rule the colony. Well, this young child, who probably about 8, 9, maybe 10 at the most, noticed by observing these ants that when these slave-making ants went out on raids to gather larvae and pupae from the colonies that they parasitized, during mating season for that ant, you'd see virgin queens join in on that raiding column and enter the colonies with the workers that were going down there to rob the brood. And when those workers came up and carried the brood back to their original colony, those virgin queens didn't come back up. So that young child had noted another way that Polyergus queens captured new colonies. That child was me. Simple observation, and you can learn quite a bit, folks. Now, when it comes to assigning numbers to these behaviors, which is what you're going to have to do, you want to rank them and weigh them carefully. For me, I give Varroa counts a few extra points. If they're down to like 1 Varroa or 0 Varroa, I rank them with a few extra points there. If it's 2 to 3, and this is per 100 bees, I'll give them maybe 5 points. 3 is supposedly, and then this is one of the gray areas about Varroa counting, supposedly the cutoff point for 100 bees is about 3 mites. Over that, you have the threshold for damage. And then, of course, anything over 3, I don't give any extra weight to because I don't want to see them go beyond that supposed threshold. So how you rank and weigh these is also kind of up to you. I'll explain a little bit more about it here on the next frame. Just do not overcomplicate this initially. Like I say, concentrate on resistant mechanisms at first that are measurable. Varroa counts, grooming, damaged mites on a screen bottom board, hygienic behavior tests, and the latest one, the cell uncapping and recapping. This is any of these things that you can observe and count are something that you can go by. And like I say, what I spend most of the winter doing is reviewing what I've went through and gathered during the year, and I'll sit down and I'll add up all these numbers that I've given them, and the colony that ranks the highest number-wise is usually my best breeder. There's one other consideration that after 26 years I figure in there, and that is longevity. If this hive that I'm looking at comes from those original bees 26 years ago, that longevity counts quite a bit for me. And I know those bees have been up and down in Varroa counts. And when you do a Varroa count and how you do it, there's so much that's gray matter in that. I'm coming to a point where less and less Varroa counts matter to me. For one thing, I have trouble anymore finding them. This spring, I took the class from my college beekeeping, beginner beekeeping class. I invited them out to my bee yard, took them out there, and I wanted to show them some Varroa. So it's springtime, there's plenty of capped drone brood. I took an uncapping knife and pulled a bunch of them out of there, and darn, we couldn't find a single Varroa. Now, like I told them, they're in there. It's just that these bees have come to a point where they've got them under such control that you have to really look for them. I told them to get on the internet and look up Varroa if you want to see one, because apparently we're not going to see any here. Next slide, please. Okay, this is out of the Bee Journal article that I did, the 3-part one. It started with the August 2018 issue. Then I had some health problems and didn't finish it till the beginning of 2019. But this lays out, that article lays it out in great detail. Probably now that I look back on it, too much detail. This may scare you to look at this record sheet and think, oh my God, what kind of work are you getting me into? Well, a lot of that on there is not needed. Like I say, what you're wanting to concentrate on are their behaviors, roll accounts, things like that, that have to do with them and Varroa coming to an equilibrium someday. You want to see lesser and lesser counts of Varroa. You want to see more and better behaviors, whether it's productivity. I don't worry too much about swarming, because later on you're going to find out that I don't think swarming needs to be tampered with. Who was it? Reverend Chadwick back in the 20s, I think. There was quite a controversy in the beekeeping world and in the Bee Journal about trying to breed swarming out of bees. And I like his answer. It's like, what are you people trying to do? Ruin the species? This is how they reproduce. That doesn't need to be tampered with. As Dewey Karen told me, this was hardwired into their genetic material a long time ago. It's become basically an instinct, and it's nothing to tamper with, in my opinion. I keep track of the hive, expansion, contraction of the bees themselves, like how many frames they're covering, what I put in there, what I take out. I keep track of my production. Any sampling or test results that I do are kept here. I keep track of the weather, other pests and diseases, like I say, their behaviors, the offspring. There's a whole listing on there of the queen cells I took from that colony and what happened to them. That really has no bearing with what we're talking about as far as breeding bees, but I try and keep everything there that I would possibly want to know or refer back to. And yeah, that's probably way overcomplicated. Just don't let it scare you. It's a future reference for evaluations and decision making, especially which queens are going to wind up being my breeder queens. Your goal is less mites, better health, longevity, productivity, and continued reproduction of your bees at the proper time. Next slide, please. As far as management goes, out with the old, in with the new is about the best way I can put it. And hopefully by the end of this, I'm going to convince you that these supersedure cells and swarm cells on the upper part of this slide are something that you don't want to take and do what's in the picture in the bottom, which is destroy them. Hopefully I can convince you of that. A lot of the management that we have done for bees is, like I say, I view it as an aberration. It's contrary to what the bees do themselves. Dr. Seeley, in his book, The Lives of Bees, lists 21 differences between human and bee management. In other words, beehives under our control and then bees out in the wild. I could add a few more to his list, I think, but I want to zero in on about 9 or 10 of those differences because they are connected to natural selection. And when going out with the old and in with the new, you need a plan of action, and especially comes swarm time. Beekeepers do all sorts of things during swarm season, and like I say, ripping out queen cells, I no longer think is the best thing to do at swarm time. This swarming instinct is the engine that drives the adaptation and evolution and natural selection and the further interaction that goes on between genetics and the environment, which has shaped most animal behavior on this planet. Basically, what we're doing with bees, once again, to refer back to Dewey Karen, is we're just engaging in population management. So let's take a look at the next slide here. Here are some of the things that Dr. Seeley lists. Out in the wild, colonies are genetically adapted to the local environment. This is something we need to let happen, and I'm not going to talk about the queen initiative in our state. Folks, if you order new queens every year from XYB or XYZ beekeeper somewhere, or somebody's lab or somewheres across the country, and put them into your hives, even though that hive's alive and you're re-queening it, you don't have genetically adapted bees. Every time you put a new queen in there from a different source, it's like you're restarting the adaptation cycle again. The queens that bees have out in the wild originated from queens in the wild. And they originated from queens in that area. There are two sides to evolution and adaptation. There's the genetic side and there's also the environmental side. And if you move an organism out of one environment into another environment that's different, it's going to have to adjust to it. And when you bring in queens from someplace else, their genes are, first off, not adapted to your area. They're in a new environment. They're going to have to get used to it. And you don't get adapted bees in just 1 or 2 years. I've heard queen sellers say, well, if you can get them through one winter, you've got adapted bees. No, it doesn't quite proceed that fast. It does proceed faster than thousands of years, but not quite that fast. Out in the wild, colonies use smaller nesting sites. If you remember back to the picture of my mentor there and think about those hives he had, basically all Edgar used was one 10-frame deep for the brood nest. And he managed to produce plenty of honey with just that one 10-frame deep for his colonies. And he had to live off of that. Other than selling honey, he did a little bit of carpentry work and did some trapping. But that honey he would take into stores and barter for other food. He had it on the shelves to sell. It was his life, and he did it, like I say, with small size colonies. To me, if you've got a colony with 3 deeps of brood, it's counterproductive. I'm certain they're using up quite a bit of the honey and nectar that they bring into that colony. So we need to think about possibly downsizing some of our hives. I mean, for one thing, you got a queen in a 3-deep hive, and she's laying all that up. She's burning up eggs or egg reserve right quick. She's probably laying 3 years worth of eggs in 1 year's time. And then people wonder why their queens fail after a year. Colonies out in the wild engage in yearly reproduction. We beekeepers try all we can to prevent their reproduction because it interferes with honey production. Out in the wild, 10 to 25% of their comb is drone comb. We tend to discourage drone comb and drones, and we will even go as so far as to remove it and destroy it. Besides not using chemicals, I don't use any of the mechanical, biological, or any other methods of Varroa control, drone brood removal being one of them. I want my best colonies to send drones out there, and hopefully they're transferring genetics to what's out in the wild. Nest relocations out in the wild are rare, at least here in the US. Now you get down around the equator and bees almost constantly will abscond or swarm, but they're living in a completely different environment than up here. Out in the wild, colonies are not treated for disease, pests, and predators. And treating them stands in the way of natural selection doing its thing. Queen and the bees will make and choose their own queens. We rely on people to mass produce them or produce them in a lab. There is fierce drone competition out in the wild, and there are multiple diverse drones that the queen mates with during mating. We select in artificial insemination the drones, and I would question that we don't get that diversity that you get out in nature by doing that. Lots of times, in order to bring about these traits like hygienic behavior, bring them to the forefront, they'll do single drone inseminations with their own brothers and sisters. Now, like one of the earlier speakers, I think we worry too much about inbreeding. That doesn't come about quite as easy, but there's one way to get inbreeding: start crossing a queen with her own brother. That may fix the trait and highlight it, but that's not the way to go. That's not the way bees do it out in the wild. They rely on multiple diverse drones. Out in the wild, bees are fed natural diet. We beekeepers tend to feed them artificially. And let me say that my recent addition, the latest hive and the honeybee that I have, I think it's 2015, they conducted tests and found that the best food for bees, like fish, is what they get out in the wild, their natural food. Lastly, I would add this too, that propolis envelope we talked about earlier, it's like a medicine chest surrounding them. We tend to discourage propolis all we can. Finally, I'll say one other thing. This text from Seeley, this Lives of Bees, this is the text that the University of Montana uses for its natural beekeeping course. There's lots of other, like I say, he had about 21 differences. Some of them I kind of question. One of them is he finds that most colonies are way up 16-foot or so on trees. I think that has more to do with what made that hole, like woodpeckers and things like that. I think bees take what are available to them. They certainly did not chew that hole in the tree. So I don't know that it's necessarily they prefer higher entrances, but that's what's available to them. Next slide, please. Once again, this worry about inbreeding, let's talk about drones a little bit. A drone is merely a vehicle of delivery for another queen's genetics. This haplodiploidy that bees have sets them apart from most of the other forms of life on this planet, where the male and female contribute completely different and unique genetics. A drone does not contribute any genetics of his own. He merely reproduces what the queen endows him with, and thus he becomes nothing more than a vehicle for delivery of those genetics. All this worry about inbreeding, here's another article for you to jot down and look up: The Honeybee Genotypes and the Environment by Maria Mexner in the Journal of Apicultural Research. All the ones that I'm listing in here are free, so you shouldn't have to pay for any of these. Simple Google Scholar search will turn these up. She points out that we should not be all that worried about inbreeding, because by the very nature of the way that bees mate and reproduce, they try and avoid that as much as possible. As long as there are sufficient drone sources in your area, both feral bees, other beekeepers, I don't think you've got anything to worry about when it comes to inbreeding. If you're in an area where there may not be a strong feral population or even other beekeepers, you can set up drone mother colonies 1 mile to 3 miles away from your apiaries. I've done that for a number of years. I have bought in the past queens from other sources. The last queens I bought were from Glenn's apiaries out in California. I bought 4 queens from him, and this is what I did with them. I created drone mother colonies out there on the periphery of my apiary to hopefully interject some of the behaviors that those bees were bred for out there at Glenn's. Wasn't too impressed with them, like about anything else that I pulled in from somewhere else in this country. None of them compare to what I've got going in my own bee yard. Whether or not these drone mother colonies and these fancy queens made any difference, I can't tell you. Only one of them lasted beyond 2 years in my area. Natural selection favors the natural reproductive behaviors that enhance reproductive success, health, and continuity. And that's why I say we don't really want to be messing around with what has been set up and hardwired into bees for a long time. And there is plenty of diversity in the US. April 2021 American Bee Journal had an article in it. I think the guy works at the Dice Lab out there in New York. They did a study on the subspecies that are currently here within the United States, and they found out that there is still quite a variety, folks, of subspecies genetics out there. I'm looking here at the article. Of course, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. There's about 20 states they never even surveyed. Some of the states, they only had 1 or 2 surveys. So I guarantee you we don't have a complete sampling of what's out there. But there is 2 members of the A lineage, 1 member of the M lineage, which happens to be the original Apis mellifera mellifera, the black bee imported here over 200 years ago, and it hasn't been imported into the US for a long time, but its genetics were found in 17 different states. Let me also say, I see this quite a bit, talk about different subspecies. There's none of them here in this country pure, folks. What they've identified are populations that have a tendency to have a lot of the genetics and characteristics of these races, but they're by no means pure. There are no pure Italians, pure Carnioans, or anything like that in this country. There's also a member of the Y lineage. There's 2 members of the C lineage and 3 members of the O lineage. One of them is an unknown. So you have 9 different subspecies here in the US. There is still plenty of genetic material out there for us. And part of what I'm trying to convince people to do is to take whatever's in your area and propagate it and keep it going. Diversity is good, and even the maladapted genes that may come from the beekeeper down the road drive selection for further adaptation and evolution. Nature selects for successful genes, and I guarantee you, if you've got a population of bees in your apiary that are being successful with Varroa and they encounter maladapted genes, they're going to get selected against and taken out. There may be an up and a down. There's always going to be an up and a down. That's just the nature of the beast. And let me tell you right now, there is no end game. There's no perfect bee that you're going to wind up with. This just goes on. And there's always a new challenge waiting in the wings out there. And Mother Nature, natural selection marches on. Tom Rinder wrote a paper about the Africanized bee here in the US. And I can find it here right quick. I'd like to read a little something from you, or for you. He ended his paper by saying this: the possibilities exist for natural selection to produce rather rapidly new ecotypes of honeybees in the Americas. The ecology of temperate as well as tropical America is rich and varied. In addition, the introduction of Africanized bees and the spread of their Africanized progeny represents an infusion of additional genetic variation. The operation of natural and artificial breeding selection on this expanded genetic variation in the varied American biomes has the potential to produce novel populations of bees very rapidly. So I'm telling you not to worry too much about the inbreeding side of this. And there is plenty of diversity out there, folks, especially if you have bees in your area. Next slide, please. Now, to get chemically free Varroa resistant bees, at some point, you've got to cut yourself loose. I have argued this with people all the way up to Randy Oliver. You can talk about it all you want. Till you do it, you ain't there. Chemicals will short circuit the natural processes. There's collateral damage to your bees from these chemicals, especially the reproductive systems have been noted as taking damage from many of the miticides and chemicals we use. When you use chemicals, you're actually helping the Varroa in a way because you're selecting for more virile and resistant Varroa. It's a byproduct of using chemicals. They're going to develop resistance to them, and that makes them that much more stronger. And like I said, you don't have chemically treatment-free bees if you're still using chemicals, and you won't get there by using chemicals. One thing that I caution you against is the use of any chemical. If you're going to use chemicals, follow that label, as everybody says. That's the law. That's the way it should be done. No chemical should ever be used prophylactically. Now think about this, folks. Doctors do not hand out penicillin to us to go around and pop a pill of it every day to prevent things. Why? Well, I've listed some of the reasons here. You're going to develop resistance in the organisms you're trying to combat. Why we think that this does not apply to bees or anything else is probably just our human arrogance. In addition to not using things prophylactically, I've seen beekeepers that engage in what I call kitchen chemistry. I do not recommend that, and I've even went as far as, after hearing some guy tout his kitchen chemistry at a beekeepers meeting, I turned him into the state. This nonsense isn't getting us anywhere. Somebody's going to interject something into their honey. An accident's going to happen. And let me tell you, the first time somebody gets poisoned, sick, heaven forbid, dies from eating contaminated honey, we're all going to be in a world of hurts. One of the worst things that ever happened, and I noticed one of the speaker from France, they don't allow them to harvest honey if they're treating. Well, apparently that's changed, and now you can use oxalic acid when you've got supers on. I think a big mistake was made there, folks. For all my life and a lot of beekeeping, the saying has been, when you've got the supers on, you don't do any treating. I think that's the wisest way to go. And while we're talking about chemicals, I see this and hear this from people. Well, I'm using oxalic acid. I'm actually treatment free because oxalic acid's not a chemical. Nonsense. Look up the definition of a chemical in your encyclopedia or your dictionary, and you'll find that an acid is a chemical substance that neutralizes alkalis, dissolves metals, turns litmus red, and typically is a corrosive, sour tasting liquid of this kind. And I guarantee you, oxalic acid, by its very name, is a chemical. So is formic acid. And I've seen beekeepers using oxalic acid. I've seen them set hives on fire. I've seen them run their bees out. I just sit back and laugh at it. I'm glad I gave all this up, and I'll tell you what, my pocketbook is glad I gave all this up, because I'm not buying the $400 vaporizer things, the chemicals and all that nonsense. I'm well beyond that. I also save quite a bit of money on purchasing queens because I don't do that anymore. Are you wanting me to still go on, James?
Yeah, you can finish. You can finish.
Okay, I was just saying because we're about to come to the most important part of all this. So next frame. Okay, this paper, or this comes from a study in Norway. I think I had the link down there, but anyhow, Terge, I hope I'm pronouncing his name right, basically did the same thing that I did. He's a commercial beekeeper though, with about 400 hives. He started out just like I did. He saw the changes in behaviors that bees were making. He kept records. He made informed decisions from the records. He would select his top 10% as breeders. He tracks measurable traits with informed assessments. And this is how he brought about his mite resistance. There are multiple traits that confer resistance. Those that reduce reproductive success are the best. The authors found that this latest cell uncapping and recapping seems to be a trait that is present in all resistant stocks and populations, fairly and managed. It's easily seen and ID'd, and as we used to say in the fish keeping world, if you can see it, you can measure it. It's a very useful selection trait. This trait is always paired with some form of suppressed mite reproduction in surviving populations. Again, both feral and managed. It targets only Varroa-infested cells, and it does not coincide with freed kill brood assays. So you cannot use that as an indication of the level of it. And there appears to be what is known as convergent evolution of this across all populations. It seems to be the one thing that is intact in all surviving populations. One thing we got to realize, Varroa, and this comes straight from Malcolm Sanford, who used to be Jerry Hayes's boss, I think, or his predecessor when he was chief apiary inspector in Florida. Varroa is now a new permanent resident of bee colonies. Its spread is impossible to control. What we need is the natural equilibrium that comes about to restore the balance that we need. Next slide, please. In nature, two things push these changes in species and population. That's the trait variation, which is the genetic side, and the environmental pressure. And these pressures either favor or do not favor certain traits. Successful reproduction is the name of the game in evolution. So these pressures favor those traits that are successful in bringing that about. The genetic diversity provides trait variation. Bee biology maximizes this diversity through those multiple matings with diverse drones. Bees also have the highest genetic recombination of any species out there. Selection pressure is necessary. I'm not all that afraid of Varroa. I don't use, like I say, even mechanical traps or things like that because the pressure is necessary. Now, I've never taken it to the level that, who was it, John Keefus and Maria Bolt did. Keefus, when he started down this path, he started developing resistance, and he saw less and less mites, so he'd go around to the beekeepers in his area and buy up their Varroa-infested brood to stick in his colonies to keep that pressure going. Now, that's somebody that's really dedicated to it. I don't think there's any need for that, but that Varroa is a pressure, and that pressure is what drives all this. Now, Turge used his top 10% for breeding. He does not chemically treat them. The bottom 30% he calls by changing queens. Those in between, he still treats. I guess if that's what you wanted to do with colonies that were not selected for breeding and maybe they were good enough to produce a honey crop or something, you could do it. When I pulled the plugs, I pulled the plugs completely. Turge does still the middle group of bees. He does treat them when necessary. The top 10%, though, he does not treat. And let me emphasize that when you start a breeding program, you do not want to treat those bees that you're using for the breeding program. You're trying to get away from the chemicals, and you've got to have the selection pressure there. Bees will interact with the natural environment and other bees. That's why they're not pets. You can't keep them confined in a tank, a cage. Maladapted genes will get selected against, and your benefits will spread into the surrounding areas. What they find around over in Europe, around the original areas where subspecies were, when you move out of that area, you hit a transition zone. It's a mixture of those genetics and the next subspecies over. And that transition zone is where things get selected for and against and work their way in and out of a population. It's perfectly normal. And that can cause some ups and downs and things, but you have to trust your bees that, and I do after 26 years, that they're going to get through it. Turge has some other recommendations: culling old comb, avoiding comb sharing. I do cull old comb. I'm not too worried about comb sharing. It could be very advantageous as long as you're sure you're not spreading diseases. He counts mites through several methods. When you do that, you need to, here's another thing with the kind of grayness of mite counts, you've got to consider the colony size. 1,000 mites in a 10,000 bee population colony is a lot more damaging than 1,000 mites in a 50,000 bee population colony. We need to learn to ID the local damage threshold, but then that's kind of all over. That's why I asked, I think it was Corey earlier, what the level is that he accepts. The 3 per 100 seems to be what has been touted. Who knows out in the wild what bees can tolerate? They've been found to tolerate a lot higher than that. He propagates his best queens. Of course, the wider pool, the better. In other words, the more hives you have to select from, the better. Remember that bees are not in a closed system. They interact with everything around them. And once again, I like people to share their experiences with other beekeepers, encourage others to try. And if you're having success, don't be afraid to write something up and send it in to the bee journals. Remember that there's never going to be a super bee that's going to solve all of your problems. However, we beekeepers, researchers, communities working together just might come up with a solution. Next slide, please. Here's this latest trait that they're talking about, the uncapping and recapping of cells. Using this kind of flow chart they've got here where they start with investigating. This can stop at any step. So it's not like it's always a complete through. In fact, it can go several ways. But it begins with bees investigating the cells. If they find one that has Varroa in it and they target it, they'll uncap them. They leave it open for a period of time and either recap it, remove the brood, any number of things. Lots of times they recap it, and this is the thing that's new. Merely opening it up and then later recapping it seems to interfere with the mite's reproduction. Well, to show I do have a humorous side, folks, I guarantee you that if I were in the midst of lovemaking proceedings in bed and somebody ripped the roof off my house, that would certainly bring it to a halt right quick. I don't know if that's what's happening there, but fresh air may come in, change the humidity, who knows what, it interrupts the mites. If you come over, I guess, can you see my pointer, James? Are you there,
Terry,
James?
Tell me what you want to point at.
Okay, move your pointer. Okay, go straight up. No. Okay, come over to the right. One more. Okay, right there. These three boxes show on the first one there, labeled A, is a sealed cell that's been opened and recapped. You can see that circular area there where they've recapped it. The middle, number B there, is a non-recap cell. It's just a regular cap taken off. The number C is a recap cell that the cap's been taken off, and you're now looking at the bottom of it. And one thing you'll note is that the silk from the cocoon that the larvae spun has been taken out with it. This is something that it's easily identifiable once you learn to recognize it, and you can count and measure it. And this is the latest trait where they interfere with the mite's reproduction, and it's the trait that, like I say, seems to be universally present in all the research that I've seen out there. So it's a good thing to select for. Okay, next frame.
I'm waiting for it.
Okay.
Got a little bit of lag going on here. There we go.
It's probably getting tired. It's been a long day.
Yeah, it's starting to sense me too. Probably falling asleep together. Not that your presentation is sleepy. That I've been going all day.
Well, to encourage everybody, we're only a couple, 3 slides from the end here.
You're doing fantastic, Terry.
Your first goal, like I say, was to educate yourself. Here's your second goal, is to create a surviving maternal lineage. Because like I say, if you go to changing queens every year, you're starting all over. In order for adaptation and evolution to occur, you have to have a maternal lineage within your environment. And how do you get there? Well, you need a queen that's made it through a winter for sure in your area. You take that queen, she becomes your breeder queen. And if you have, say you have 500 colonies, you may select more than one breeder queen, but you want to select your best queen that's made it through at least one winter. That spring, and this is my take on it because there are other things you could do from what I'm about to tell you. But what I do is the next spring, when swarm cells are made, I take those swarm cells and start new colonies, and those daughter queens are then evaluated throughout the year. The best will eventually become next year's queen source. Evaluations don't necessarily end that first year. You overwinter them. You have another window of evaluation, and that is the spring buildup up until the times they start making drones. Because if there's any problem with a colony and you don't want its genetics to go forward, that's your last chance to get rid of it in the spring before they make drones and send them out to do their thing. You keep doing this, keep judging them through the traits and the records you're keeping, the mite counts. You isolate the best queens. Those are your breeder queens. Once again, you don't want to use mediocre when you're using a breeding program on anything. Mediocre won't get you there. You continue this process, there's no in-game perfect bee. By eliminating the least fit, the environmental forces are going to help adapt a species to both the biotic and abiotic factors that it encounters. Natural selection will refine both the host and the parasite's adaptations further as the process continues. Natural selection is not going to say, oh, honeybees, I got to favor them over mites. No, it's working on the mites too. This is always going to be an ongoing process. Now, the thing where I differentiate from a lot of people is I strictly anymore use swarm and supersedure cells. I no longer create emergency queens. And now I'm going to go into a little bit of rant on that, why the emphasis on swarm and supersedure cells. It represents successful reproduction in an environment. And like I say, that's what natural selection is all about. They've been doing this for millions of years since diverging from their solitary wasp-like ancestors. The species that we call Apis mellifera is identified by several hardwired instinctive behaviors brought about by macroevolution a long time ago. These include comb building, honey storage, modified ovipositor for defense, their vegetarian diet, and swarming as their mode of reproduction. This is a hardwired instinctual behavior. These are types of things that I don't think we should really be messing with. Now, there's another process that evolutionary biologists recognize besides macroevolution, and that's microevolution. It proceeds through a process called gradualism, where gradually small changes will bring about greater changes. This is basically what happened when subspecies diverged. They settled into an area. A lot of them were caught off probably during glacial times, and they sat there and began through this gradualism process to evolve and adapt. These small changes can bring rapidly out of necessity big changes. And as Ed Wilson has pointed out with insects, those changes can come about very rapidly, especially in a new environment or if confronted with a new parasite or disease. And the differential success that they have in reproducing gets filtered by environmental factors. This creates what is known as a bottleneck, and it advances the successful and favored genetics over the unsuccessful. Natural selection advances. And I agree with Wilson too, that the fate of natural species and selection should not be left in the foolish hands of humanity. Let them do their thing. And once again, remember that there is a difference between domesticated pets and organisms that are still directed by natural selection, and that is the category that bees fall into. My feelings have always been that swarm and supersedure cells probably are the best representation of what bees have when it comes to advancement and natural selection too. It's the successful accumulation and passing on of behaviors, characteristics, genetics, traits through the generations, and through this hardwired instinctive process that we call reproductive swarming. I personally have thought for a long time, because I spent a lot of time studying ants and wasps before I ever came to bees. And I thought back then, you know, those eggs that the queens lay for ant colonies and wasps, they all pretty much look the same, and there is no special cell constructed for new queens. As far as we know, there is no special diet for new queens. So how do those eggs differentiate into workers, queens, drones, and oftentimes with ants, there are even other castes such as soldiers, seed crushers, things like that. Well, I've often suspected that there is such a thing as what's known as a queen egg, the egg that's destined to be a queen from the beginning, the ones that get deposited in queen cells and supersedure cells. I think here, yes, this How, and I don't know if I'm pronouncing their names right, sorry guys, this paper, A Maternal Effect on Queen Production in Honeybees, lays out and kind of adds support to what I'm saying. Traditionally recognized ways that queens get differentiated are by the diet, the special cell, the worker attention, they dance, cage, comb themselves, and the effects of other queens. These people in this paper have found, though, that the eggs that queens lay into queen cells and supersedure cells that are destined to become queens from the beginning are larger and heavier. They only occur during the reproductive season. They are marked by a unique set of pheromones. There are other in ovo effects. The queen morphology and physiology of the queens raised from these eggs, there are several significantly differentially expressed genes that involve hormonal signaling, body development, immune pathways, key traits for differentiating queens and workers. The queens have larger ovaries, they have greater size, weight, and greater fecundity. All of this indicates what I've always suspected, that there are maternal effects that those eggs get from the queen. And this paper helps prove that. And it shows that those maternal effects in the queens have a far more active role in the queen's production of genetic material in the next generation of queens than previously thought. If you want to reap the full benefits of what bees are trying to do, I think this is where you'll find it. They go on to say they did comparisons with with emergency queens and the like, and the queens from the queen cells always did better. This is my argument for using swarms, supersedure cells, and supportive selection as a sustainable way forward by allowing and encouraging with this selective stock improvement, both natural selection and the bees to do what they do best by the beings best suited to them. Next frame, please. Once you start getting colonies that are surviving, you have lesser and lesser Varroa counts, your bees are on the way. And here's the third goal that I can lay out there for you: break the old bee-eater myth. For years it was said, don't treat your bees and within 3 years they're going to be dead. Well, as soon as you break that myth, you're headed down a different road, folks. Your bees are on the way. Every year after that becomes another goalpost. Once again, remember, there's no end-game perfect bee. Those people that breed those fancy fish don't stop when they get someplace. It's constant selection onward. You need to also realize that hives need maintenance. You need to take care of the old comb needs to go, the work that needs to be done. If mice chew a hole through the back of it, you need to replace it. Colonies with longevity is nothing new either. Langstroth in his book noted colonies that survive for 20 to 40 years through requeening themselves. So there's you another goal with breaking this 3-year myth. Let's go to the last slide here. here's a summary basically of this process, and I will give you my email or have James post it should anyone have further questions, but I've tried to make it as simple as possible here. Like I say, you need a surviving queen to start with from the breeder year, your breeder queen. This is going to be the start of your material line. Use queen cells to make increase. I ran this by a few people. One of them asked me, what if my colony does not swarm? Can I split it? The researchers that found out about the maternal effects also found out that if you raise queens by the emergency route, allow the workers to choose the queens. This kind of throws grafting out the window, things like that. Anyhow, evaluate your colonies, your queens, test them, record, call in the fall. I'm a big believer in take your losses in the fall, make your increase in the spring. And then the next year, use those evaluations to make decisions, choose your new breeder queen or queens. Continue the process, you should see a drop in mite numbers, better health, increasing productivity, less losses, better beekeeping. And that's where I'm going to end that. And James, you want to put out my email or?
Yeah, Terry, thanks a lot, man, so much. We appreciate your presentation. Your email was posted in the chat, so don't be surprised if you get a barrage of questions and inquiries, which I'm sure you'll be happy to field.
That's fine and dandy with me.
You're an excellent teacher, and we really are benefiting from your expertise. I can't say or express how grateful I am for having access to your experience as well as the things I've learned from you already.
Well, I'm very glad to hear that. I'm glad to see you put on this conference, and I'm glad to hear that you are with your group there in Michigan, considering training beekeepers around the Boy Scouts requirements. I'd also throw out there to you that the British Beekeepers Association started requiring beekeepers to get certified and go through what you're proposing with that. At first, it was not met too well with beekeepers, but interviewing them recently, they say it's one of the best decisions they ever made. They are winding up with a better caliber of beekeepers through their training program.
Yeah, I might go out on a limb to say that a lot of loss could be mitigated by better beekeeping management practices. And I say that experientially just from what I've suffered through, and also some of the gains. And I just, like, more and more each day, I find myself questioning, what do I do now? I thought I had a plan, and I enacted that plan, but was it the right plan? And that's one of the beautiful challenges of beekeeping is always being presented with some new dilemma to resolve. So
yeah, no matter if we do solve the Varroa problem tomorrow, there'll be something else come along.
That's right. All right, so Terry's email is in there. We didn't get a whole lot of questions because you sure offered an awful lot of information for people to digest.
That's why I tell people not to be afraid to get a hold of me. I realize it's a lot to digest and packed into a short amount of time.
Well, and that's the thing. You get a lot of compliments from people who've been doing it a long time, and people who have done beekeeping for a long time are even learning stuff today. So, real quick, if you could just, Mark had a quick question about when you first started out, what your colony losses were like, what you experienced.
Okay. Yeah, that actually is in that article that I published in the Bee Journal, but I'll refer back to it here. I started this process in the spring of '97. And the following year, I had an 83% loss. And this is when I pulled the plug on all the chemicals and started letting them raise their own queens. Next year, it went down to 78. 98 to 99, it was 25. 99 to 2018. 2000 to 2001, it went down to 21. So those first 5 years averaged out to 42% loss. But I can't say that it got better because not too long after that, I think one of the new forms of deformed wing virus came through and losses went back up again, but then they leveled out. Anyhow, you come to the last 5 years here, and I'll include this year because I was just out today at the hives, and every one of them, they're zooming in and out. They're bringing in maple pollen. I'm quite certain they're in good health and they've made it. So starting 3, 4, 5, 7 years ago, I had a 10% loss. Next year, I had 0. The year after that, it was 0. The following year, I had a 3% loss. Then I had 2 more 0 years, or no, then I had a 0 year, a 5%, and a 0. And for me, I only maintain about 30 colonies. So these 5 and 3% are basically just 1 or 2 colonies being lost. One thing I didn't put in this article, I wish I did, and I think the person you're talking about asked about it, Varroa counts. They were wild in the beginning. Nowadays, I have trouble finding Varroa. I mean, and I'm to the point where, you know, there are some of the bee wazoos in the world may want to knock me for this, but I'm about ready to say to heck with Varroa counting. You know, I've got colonies that have lived 26 years. They're doing just fine. I'm not having any problems. Why do I want to get in there and disturb the bees more with Varroa counts when I'm not finding any? I know there's some in there. They're in every colony, but I think the bees have them under control. And I'm about to the point where I'm ready to give up on all this Varroa count nonsense. Takes up too much time, energy, disrupts the bees. I no longer see a need for it with me.
Well, Terry, you definitely are providing us with an excellent example for us to follow and to maybe even replicate in our own apiaries. You give us hope that it's possible. I'm so grateful again for you to come today. Thank you so much for your presentation.
To 2018, 2000 to 2001, it went down to 21. So those first 5 years averaged out to 42% loss. But I can't say that it got better because not too long after that, I think one of the new forms of deformed wing virus came through and losses went back up again, but then they leveled out. Anyhow, you come to the last 5 years here, and I'll include this year because I was just out today at the hives, and every one of them, they're zooming in and out. They're bringing in maple pollen. I'm quite certain they're in good health and they've made it. So starting 3, 4, 5, 7 years ago, I had a 10% loss. Next year, I had 0. The year after that, it was 0. The following year, I had a 3% loss. Then I had 2 more 0 years, or no, then I had a 0 year, a 5%, and a 0. And for me, I only maintain about 30 colonies. So these 5 and 3% are basically just 1 or 2 colonies being lost. One thing I didn't put in this article, I wish I did, and I think the person you're talking about asked about it, Varroa counts. They were wild in the beginning. Nowadays, I have trouble finding Varroa. I mean, and I'm to the point where, you know, there are some of the bee wazoos in the world may want to knock me for this, but I'm about ready to say to heck with Varroa counting. You know, I've got colonies that have lived 26 years. They're doing just fine. I'm not having any problems. Why do I want to get in there and disturb the bees more with Varroa counts when I'm not finding any? I know there's some in there. They're in every colony, but I think the bees have them under control. And I'm about to the point where I'm ready to give up on all this Varroa count nonsense. Takes up too much time, energy, disrupts the bees. I no longer see a need for it with me.
Well, Terry, you definitely are providing us with an excellent example for us to follow and to maybe even replicate in our own apiaries. You give us hope that it's possible. I'm so grateful again for you to come today. Thank you so much for your presentation. I'm going to go ahead and give everybody some following closing remarks and some follow-up comments, and then wish you and bid you all a good night and a good evening so you can get some rest, because I know that I'm tired. My brain is full, but my body's exhausted. So it's been wonderful.
My stomach is empty too.
Oh yeah, yeah. I didn't think about a dinner break. I figured we could make it all the way to the end. So let me finish up and conclude with everybody by giving you an overview of the Sustainable Beekeepers Guild of Michigan. Facebook group is open to everybody. It is an open forum. You don't have to be a member of the club to participate in that forum. We also have the Facebook group for the Michigan treatment-free beekeepers. It was a little bit of a different posting and conversation policy between the two groups, but the reason for that is clear and laid out in those policies on those particular venues. We want to remind you about some of the efforts that we're hoping to aim for with our queen breeding and nuke exchange program for beekeepers. We want to be able to establish a network and a co-op of beekeepers that are practicing a lot of the things that you're hearing and experiencing today, so that others can benefit from the genetics in the region and also improving the genetics in your region. I like to say I'm not an altruistic beekeeper. I want you to have better bees so that your better bees are helping my bees be better also. I probably will never say that the same way twice. We are also working on a mentor group network development. You know, we want other beekeepers to have access to mentors who are, you know, providing treatment-free and chemical-free practices. And it's not just the chemical-free, treatment-free aspect of beekeeping, but you're learning basic hive management. You're learning what you need to do when it comes to splitting. You're learning about bee biology, and you're getting hands-on frame time. And then Terry had mentioned that we're discussing how to create a hive or badge program for beginners. We want you guys to be able to earn a hive. You know, the Bee Guild has a plan for you to complete these processes, and at the end of this period of apprenticeship, you not only get the badge of honor of having had completed that, but we'll provide you with your first nuke. We'll give you your first set of bees, and we'll give you some good bees. All right. We also want to encourage you to reach out to us. Send me an email, send me a text, whatever you need to do. Stop by the webpage for the Guild, shoot us future speaker and topic recommendations. And the last thing I wanted to cover is that we're working on a forum for individuals to come together and share, other than the Facebook venue, not that that's not sufficient, but a sustainable beekeeping guild where people can come together and they can express their views and opinions, and they can share like-mindedness in these efforts without having to face the barrels of some of the more mainline forums where you can easily get distracted or even harshly treated for just having the idea or notion that it's possible the bee could take care of itself and its genetics. We want to be able to give people that place and venue to learn from each other on a consistent basis. We don't have to just wait for conferences and meetings to get that information. You can go right online, go into that forum and share that info with each other and have access to each other. With that, I thank you all so much for those who have stopped by. I thank you all so much for hanging in there with me, and to the last 20 or so participants that are just there by a thread. Have a good night. Enjoy your spring, and we'll be talking to you soon. Good night.